And the next session is on WMP and plant and animal microbiomes, where we have Rebecca Stumpf, professor at University of Illinois in the US, Tim McAllister, Agriculture and Agric-Food, Canada, and Carolee, professor of plant pathology and systematic bacteriology at Pennsylvania State University. So presenting. Thank you.
Good afternoon. I guess it's still morning. It's really a great honor to be here, especially as this is the inaugural summit of the WMP. My name is Rebecca Stumpf. I'm a professor at the University of Illinois. I'm a senior editor of the journal Animal Microbiome. Most of my research has been based for the past 30 years in Equatorial Africa, working on primate comparative microbiomes, and more in the last 5 years or so doing more research on infectious disease and surveillance. This talk is a collaborative effort with my colleagues, Dr. Carolee from Penn State, an expert on plant pathology and the microbiome, and Dr. Tim McAllister from Alberta, Canada, working with the Canada Agriculture Agri-Foods Agency. Unfortunately, they were not able to be here today, but they are, I believe, online for questions, and they will be here today online. And we have also put together this integrative talk where they have done some pre-recordings so you will hear from them directly.
Microbiomes are everywhere. They're diverse and they're evolving. We've heard about the microbiomes that associate it with humans and how it has a major impact on our health, not only from the perspective of our digestive health, but also on our mental health because of many of the metabolites that are produced by the microbiomes that then can have secondary physiological effects on our body. We find microbiomes in the decomposition of manure and during composting as well as a microbial-driven process with a variety of different microbiomes. Microbiomes can affect our respiratory health. We have a respiratory microbiome. And in the cattle, we have the rumen microbiome, which is involved in the digestion of the plant material that they consume and encode for the genes that are unique in terms of their ability to break down plant cell walls. The microbiome of the soil is also incredibly complex and contributes to soil health and the exchange of nutrients between plants, insects, and the broader environment. Plants may have both epiphytic and endophytic microbiomes, something we'll hear about more later. And of course, microbiomes can also impact water quality. We hear about the potential of microbiomes in the form of biofilms being sloughed into water, transferring and moving in the water stream and the water column, which can then cause contamination of the water if they're pathogens that are entering the water system. And then microbes also play a major role in nutrient cycling and uptake, as well as in biogeochemical and chemical cycles, such as the carbon cycle and the nitrogen cycle. All of these are largely driven by microbiomes within these various environments.
Plants and animals have interconnected microbiomes, and the microbiomes from managed and natural or wild ecosystems are also interconnected. The soil is a particular driver of that interconnectedness. It is a diverse ecosystem of many diverse microorganisms that serve really as a repository for the microbes that make up the animal and plant microbiomes. Luckily for us, when discussing animal and plant microbiomes, we can rely on host microbiomes with similar principles, or the fact that host microbial interactions or processes are common between plants and animals. For example, there is microbial specificity that occurs in both. Not every microbe will colonize every host or every part of every host. There is coevolution between the host and the microbial communities that are colonizing them that have real fitness impacts for both. there are functional communities that can be described for each of these niches. So although there's so much to talk about investigating across these hosts, we found common research themes in our discussions. For example, microbe acquisition. Is there microbial or host selection for this process? Or does the host microbiome serve as a holobiont or not? These are just some of the common research themes across animals and plants.
In my laboratory, we spend a lot of time studying cattle microbiomes. So a single host can contain or possess a number of different microbiomes. We've looked at the oral cavity or the mouth microbiome. We've looked at the esophageal microbiome, the respiratory tract microbiome, the digestive tract microbiome, and even along the digestive tract, if you look at the rumen versus the lower intestinal, small intestine, large intestine, we'll find that the microbiome differs along the digestive tract as well. There's also the skin microbiome and the vaginal microbiome. These two areas may have a major impact on determining the nature of the microbiome that establishes in the offspring as they come in contact and are transferred to the offspring either during or post birth. Now, these microbiomes can also have, when things go awry and there's problems with the microbiomes, they can lead to a variety of different diseases, such as bovine respiratory disease and the impact that the microbiome has on it. Bloat and acidosis are conditions that result from improper functioning within the rumen. Displaced abomasum, liver abscesses as well. We can have, in some instances, microbiomes form on the surface and form liver abscesses in the liver itself. Mastitis, metritis, or reproductive tract infections, foot rot, all of these are mediated through microbiomes, and in many cases where things have gone wrong with the microbiome, the balance of microbes within those various areas that have led to disease. This is all influenced as well then by the nature of the host's immune system, which may impact the bacterial and other species, microbial species that are associated with those microbiomes, no matter where they lie within the body of the host animal itself.
When we think about plant and animal microbiomes and the research that's been going on in the past 20 years, often we are conceiving of livestock, and that's where most of the research has been focused. But what's been going on in wildlife microbiomes in the last 2 decades has also been incredibly powerful in providing, as you've heard from Jim and some of the other speakers this morning, a really great comparative perspective that is not necessarily as available from domestic species. This diversity of hosts, these natural systems, this very powerful way of looking at these different systems has provided enormous information and has enormous potential for these biorepositories and other comparative perspectives that give us incredible insights into functions and diversity. In particular, primates, which are our closest evolutionary relatives, they have provided a lot of insight into the uniqueness of the human microbiome. The vaginal microbiome, for example, in humans is unique compared to all other wild primates. The other aspect that's important with wildlife microbiomes, as has been mentioned, is this fact that more than 70% of emerging diseases are of zoonotic origin. So understanding more about where these are coming from, their surveillance studies, is such a critical area of research. As Jim also mentioned, this reservoir, and some others of you, this reservoir of AMR in these wild populations in the oceans, etc., are really an unknown potential direction and an encyclopedia of new drug discovery, etc. So applying these kinds of, I'm not sure if I'm the one who is, yes. Applying these really powerful comparative perspective, taking Jim's human biogeography of the human microbiome body sites to a broader perspective of different species, host species, is another fundamental area of research that's critical. Looking at archaea, viruses, bacteria, fungi, parasites from their evolutionary histories and comparative histories can provide us a lot of insights into human health and animal health as well. For example, one of the things that our group has found out in the last few years is the real conservation of the oral microbiome compared to all other body sites that's conserved amongst all primates. So this is something that that comparative perspective has been quite helpful in elucidating.
Some of the top research topics that we discuss when talking about phytobiomes are really some of the same things that are discussed with animal microbiomes. But we are particularly interested in the acquisition or transfer of microbes from one generation to another, or from the soil into the plant phylosphere, or into the endosphere. We know that nutrients in the rhizosphere attract certain organisms, and that fast-growing organisms will grow and multiply in the rhizosphere. And there's a subset of those that make it internal into the plant or are on the phylosphere. Although there's vertical transmission through seeds, that can happen, these organisms that are carried from seeds from one generation to another, or environmental inoculation from insects, for example, or other external features. There are a small number or percentage of the microbes that are present in the plant. And the soil really does provide an inoculum for the plant. The endophytes that colonize the internal tissues are important. They can be both beneficial or pathogens. We discuss disease suppression of microbes by other microbes, right? So the pathogens are suppressed. And we're working certainly to develop microbial inoculants from these study of disease and disease suppression. Another hot area of research are interest in core microbiomes among different either plant types or families of plants or niches within the plant itself. Phytobiomes encompass the interaction between plants, their environment, soil, air precipitation, insects, and other visitors, and their microbiomes. And it's the emergent properties of these phytobiomes created through interkingdom interactions that we try to study. Although it's difficult to study because the phytobiomes are not just the sum of their parts. These properties are changing with the intensification of agriculture as well, which makes it difficult. And as in many microbiome studies, although phylogeny sometimes approximates traits, and we can look at traits related to taxonomy, sometimes we have to get straight to the genes to understand what the functions of these phytobiomes are. Although synthetic communities are a major target of work in phytobiomes, developing them and using them are very difficult, and it is difficult to commercialize these complex communities, especially if they have disease suppression qualities. The other real topic that is difficult for us to research at this point is looking at causal agents of particular phenotypes within the microbiomes, and developing a set of Koch's postulates for microbiomes is critical.
So Tim and Carolee and myself, we came up with basically these real key knowledge gaps that was interesting amongst the three of us studying these different systems that really led us to this, that there's so much synergy in what these knowledge gaps are. Largely, we're at the tip of the iceberg for many of these, and much more understanding and knowledge and synergy amongst these different disparate systems is going to be the way we think moving forward. Recognizing microbial diversity, the mechanisms and the functions of these microbes in all different systems: soil, aquatic, insects, wildlife, natural areas. What are the factors determining the composition of the host microbiome? Niche specificity, host biogeography, exogenous factors such as the habitat, diet, sociality, social factors of the hosts, impacts of these microbial communities, these consortia that we find in these different disparate systems, host behavior and the microbiome. What's driving these behavioral differences? How does the microbiome impact these behaviors, this gut-brain axis that remains such an intriguing area in humans as well as other species, looking at aggression, looking at anxiety, autism, all of these different, and then of course the treatment implications of this. Host microbial ontogeny and acquisition. This is a key element of research, as some of you have mentioned. How are these microbes acquired? How is the host immune system specificity impacting that? Horizontal and vertical transmission, we still need a better handle on this. Microbial community change through development. How does the host microbiome in these different body sites, in these different systems, change from incipient birth to death, through to death? These devomes, host microbial signals during development, they're so critical to immune function and host development. Some of the other knowledge gaps and key areas of research, of course, are in particular with One Health, emergence of novel pathogens, these pathogen transmission routes and factors influencing transmission, we know is a key area that needs further study. The virome, we are at the very beginning, or not the very beginning, but this is still a really difficult research topic due to the quickly mutating genetics. Prebiotics and probiotics, of course, those of you in drug pharmaceuticals have a lot more to say about that, but that is a clear health direction. Microbial transplants, the same thing. In humans, livestock, wildlife reintroductions is also a consideration. Passaging in plants, suppressive soils that Carolee talked about, and of course AMR, and the potential for new drug discovery. And the factors driving or impeding horizontal gene transfer. Can we make predictive microbiomes, and how resilient are they? Can they be manipulated? What are the success rates? Clear areas of need, not only in animal microbiomes, wildlife microbiomes, and plant microbiomes as well, and humans. The changing climate, how does reduction in microbial diversity, what does that look like? How can we maintain diversity? Also thinking about bioreparation, remediation of microbes in advancing water quality, water treatment, plant and animal overall health through climate change. All areas of key focus.
There are also a number of technological considerations, such as the microbiome sampling. What is the volume that's going to be sampled, and how representative is that of the environment that it's being collected from? Site considerations as well. Where within the environment are you going to collect it from? And temporal considerations. What time are you going to sample at? Time of day, diurnal effects can influence the nature of interpretation of the microbiome. Also, then we move on to the extraction methods, DNA, RNA extraction methods. What's the yield of genetic material? Is it sufficient to sequence? What is the quality of that material and its stability? There are a number of nucleases that can break down the DNA and the RNA. And are there inhibitors that may influence and interfere with the overall sequencing process? Then when we move on to sequencing, we have to think about what amplicons we're going to use versus shotgun sequencing, and whether we're going to use what indicators we're going to use for bacterial or microbial diversity, and what regions are we going to amplify within those indicators. The platform, whether it be long or short read sequencing, can also have an impact, and the depth of as to how much depth we are in terms of sequencing that genetic material, in terms of the number of times that we're going to sequence it over. And then the cost as well has to be considered. Even though the costs have come down considerably, they still can be quite significant if you're looking at trying to interpret complex microbiomes and complex environments. Then the bioinformatic analysis, there's always the need for some processing and cleanup. Sequencing is absolutely perfect. And then the pipelines as well that are available for interpretation of those sequences. When we get into that, then we look at sequence assessment, sequence function, and then steps to further strengthen those databases so that we can enhance our interpretation and characterization of the microbiomes that we're attempting to define.
Where do we go from here? Of course, we need better technical and analytical methods. We are thinking about how to counteract the reduction in diversity and practices that depopulate microbiomes in all ecosystems. How do we take a holistic approach to diversification or optimization that goes beyond inoculants? Predation is an area that has received very little study, and it's so important for microbial turnover and for the interactions that we have been talking about thus far, and deserves more attention. And all of this is really set up so that we can do translational microbiome sciences for every ecosystem.
Of course, we also need to pay a lot of attention to inclusive science, increasing inclusive science, increasing global South perspectives, increasing global South training, capacity building that will lead to better science, and that we think is also, of course, part of WMP's ability to advance this.
So this leaves us with what is the role of the World Microbiome Partnership? We know that studying microbiomes is incredibly complex, so knowledge generation is going to be a key component, combining expertise and funding from different countries to increase the impact of microbiome science. We need translation, where we translate microbiome science into opportunities for industry and the public. We need to inform practice so that we can extend the nature of microbiome science to stakeholders, to encourage adaptation of where the information can offer the greatest benefit. We need deployment and aid in aligning regulatory programs to be applicable to complex microbiomes. As it is now, many of the regulatory principles that are applied really aren't relevant to complex microbial populations like we encounter in microbiomes. We need data harmonization and sharing, increasing visibility and adherence to best practices, clearing house for approaches for agreed consensus, acceptance, and updating and training, so that we can get a common feel for microbiome analysis and how we interpret the function and roles of microbiomes within the various environments from where they're derived. Combining all these factors together will make the overall microbiome group and the program be able to deliver as much as possible from a One Health perspective to the benefit of science and humanity as a whole.
Carolee, Tim, and I just want to thank the organizers of the WMP, Joel and Lida and Claude and the country of France for bringing together a very transformative and synergistic approach that we really think can make a big difference in advancing microbiome science.
Thank you very much. I don't think we have any question online, but it's open for discussion in the room, and we have time.
Oh, well, thank you. A question about how are you, and how might you engage zoological societies, zoos, as you think about wildlife, obviously in a more captive environment, and beyond zoos, greenhouses, and botanical societies, are they being engaged, or could they be engaged further in these efforts, both zoological and botanical societies, in understanding the environmental and animal microbiomes?
That is an excellent question, and it brings up enormous need for that. Where I work in Bugodi, Uganda, my partner there works with Unite, which is a North Carolina zoo organization, sponsored organization that does provide teacher training in conservation and environment in rural Uganda. That's been an enormous help in advancing and training and focus and working with young children in conservation and animal health. But that's just one, and I fully agree that needs to be more developed, and figuring out frameworks, working with local communities who have already done this successfully, like my partner there in Bugodi and Unite. It would go a long way to getting these partnerships together and advancing training. It's a good point.
Yes, Gianfranco?
Yes, so this is probably a question for the two sessions, because it's very difficult to separate humans from animals. So it's very fascinating. My question is more on translational, because if we talk about translation of all this knowledge and science, we are still very far on what could be a therapeutic use of the microbiome. I know much more the human situation, where probably there are less than 10 phase 3 as a clinical translation of the knowledge of the human microbiome into therapeutic approaches. So maybe the animal or the plant microbiome could help us to bring more information about that. And also, this is one question, so what do you think about this? And the second question is more, since we don't know what is a healthy microbiome, or it's extremely difficult to define what is a healthy microbiome, of course we talk about diversity, and this is kind of a general statement. How do you think are the main challenges now to better know what's going on in terms of health?
That's an excellent question, set of questions. Carolee or Tim, I wonder if you want to, I can add to that as well, but I wonder if you want to start.
Yeah, well, I can say a little bit. Yeah, I think like when it comes to food production, if we're talking about food producing animals, you're right, we're at a very state of infancy in terms of our knowledge. A lot of what we're focusing on now is just changing production practices to manage what we know about the microbiomes. Of course, the areas of probiotics and the potential to manipulate those microbiomes is definitely an area of interest. But we've found that they're fairly robust and not easy to change in terms of the direction they're going. So when we introduce other bacterial species into the most microbiomes, often they fail to establish or to have a great impact due to the robust nature and the repetitive nature. There's a lot of common metabolic pathways shared by multiple species of organisms. That may be an adaptive mechanism. If one pathway fails, another pathway can still continue to function and contribute to the health of the animal. And then what leads to the dysbiosis and the disease in the animals is another main area of study.
Yeah, and I can say a few things as well. For the plants, we have used microbiome sciences to develop products that are now on the market. There are some companies who are specifically working on things like drought tolerance, and they've been able to harness the understanding of the microbiome, especially related to in a drought situation. If they find a plant that's doing very well, then they can look at what is that microbiome compared to those plants that aren't doing very well. And they've been able to harness some organisms that they now
The magics of internet.
Well, Carolee's, it's 3 a.m. over there, by the way. So, but I also, while she's waiting to connect, drug discovery.
worldwide benefit.
So we lost you for a few seconds, 30 seconds, something like that.
Carolee, do you want to repeat the last your last point?
Yeah, so
Okay.
part.
Yeah, it's good now.
And so working with prebiotics, really looking at what management practices in plant health is important.
Okay, well.
Yeah, I'll just
Okay,
quickly say a couple points about AMR. There's so much that we, like was mentioned with the oceans, with the wildlife, with natural areas. There's so many AMR genes there that we've never seen before. And these are in a really important area of understanding patterns, understanding these mechanisms of how resistomes and resistance work can lead to different ways of applications, yeah.
Just a quick follow-up, Joel. I think so important. A lot of attention on the resistome, and I mentioned antimicrobial peptides and the marine work from China. But this could be very beneficial, right? And understanding that resistance,
Yes.
natural antimicrobial resistance, could help preserve microbial biodiversity.
Yes.
And how we could foster that
Yes.
could be, thinking beyond human, could be very important.
Yes, especially moving forward with climate change and all. Yeah, that's a very good point.
Yagalem?
If I could just add one quick comment with that last question. Really, this comparative perspective can give us a lot of insights into applications, because if we are trying to develop drugs that are so monoculturally focused, that's going to be a problem, because that diversity, these microbiomes in the immune systems and that relationship is can be so resilient to change. So this is something.
Thank you. I just want to know the one element, is the socio-economic aspect of these microbiomes. Impacts that we can just identify, that just has already been identified as best practice, or something that we can take as a lesson so that we can adapt it in our local systems, if something that you would like to share.
Do you mind repeating the question?
The socio-economic aspects of the microbiomes, particularly in the human health system or ecosystem, that we can share, or the impact that we have already identified and share it with any of the stakeholders, so that we can adapt it in our local system.
I guess,
It's all about impacts.
Limitations to translation to humans, into applications, really?
Are there limitations to translations to humans? That is what you're asking? Oh gosh, yes. Are you talking about ethical limitations, or you're talking about scientific
Impacts, the outputs, or best practices that we can share, or lessons that we can just share with any of the stakeholders whom we are considering in this platform.
Okay, Tim and Carolee, do you want to respond?
Well, I could make one suggestion. We're and Caroline pointed it out as well. Like we're increasingly showing that microbiomes have a key role in both plant and animal health and productivity. And when we talk about that in the agricultural sector, we're talking about food production. So in terms of the contribution of microbiomes to food security and the implications that has for both social and health and well-being of humans is critical. We got lots to learn yet, but we really see, in terms of future progress, the microbiomes are a key area in which that progress is going to occur.
I guess Yagalem, addressing that question from an Ethiopian point of view, maybe addressing the question of the potential for translation everywhere in the world, and that might be a socio-economic burden or difficulty.
Yeah, I would just say one thing about that, which is translation and application and access is an ongoing concern, especially for those in the global south who have not had free access to vaccines and things like this, and just emphasizing how important training and research driven by the global south is in advancing their own perspectives and methods and drug discoveries that will be advantageous globally, but also locally.
Yeah, I think we'll leave it here. Emmanuel is suggesting that we do have references, we do have standards, we do have a lot of things that can be shared in terms of technological advances at the moment. But truly, we're entering the socioeconomic aspect, and so there's more to do probably. Just one comment on my part. You mentioned inclusivity and capacity building. I think this is an extremely important aspect. We had some difficulties actually to attract broadly for today's event, and I think we have to work more on that. So if you have suggestions, we welcome those really sincerely.
Happy to.
Thank you very much.